Good evening. My name is Greg Beidel, and I am the Associate Director for the Center for Genetic Medicine here at Northwestern University. It is my pleasure to welcome you to tonight's installment of the Silverstein Lecture Series. This lecture series is made possible by a generous gift from the Herman and Bea Silverstein Foundation for Medical Research to Northwestern University and the Center for Genetic Medicine to promote awareness to the public of recent advances in genetics and medicine. And tonight we we do this by bringing in distinguished and illustrious speakers from all over the country and the world. And tonight we have an extremely interesting speaker, Dr. Lou Guillette, and his talk, I think, is going to be unique in this series, and I think of it kind of as epigenetics meets alligator wrestling. And I'll let Dr. Guillette explain that more. But just to give you a little background on Dr. Guillette, he is professor of obstetrics and gynecology at the Medical University of South Carolina, and in a very unusual combination, he is also an endowed chair in marine genomics of the Hollings Marine Laboratory. And that kind of combination is extraordinarily rare, but speaks to the different worlds that he is bridging. I won't go through the long list of awards and positions that Dr. Guillette has held. Let's just say that he's published more than 300 papers. He's edited 5 books. He's been a professor with the Hughes Medical Institute, and he's served on committees both for the United States government and foreign governments on environmental impacts of endocrine disrupting chemicals, and the list goes on. But I won't. So I will just turn it over to Dr. Guillette and say we are pleased to have you here, and we're looking forward to a terrific talk.
Thank you all. It's a great pleasure to be here, or I should say it's a great pleasure to be here again. We're trying to do calculations. Kelly Mayo and Teresa Woodruff brought me here, I think about 5 or 6 years ago. And so it's great to be back. I'm not sure if it's great to be back for your weather, but that's a different story. I left Charleston this morning and it was already 69 degrees and it was supposed to be like 77 and blue skies today. So our azaleas are already come and almost are gone. And so spring is well sprung where I'm at. In fact, I'll be complaining next week when I'm in the field and it's in the 80s. So I'll be thinking about you all. So what I'd like to try and do tonight is something I think a little bit different. One of my students actually tried to describe what we do in our lab, and it was kind of like Evo, Devo, genetics, repro, and it just went on and on. And I just say we're reproductive biologists. We're interested in the reproductive system. For me, in my position, I'm interested in healthy babies, and I'm interested in healthy baby wildlife. And so hopefully you'll see that here tonight. So first, I'd like to thank you for inviting me. It's an honor to be here. And so, like the little guy here, I bow to all of you and thank you for being here. And of course, this sets off this whole lecture because, of course, he's bowing to her for a very specific reason. So, a couple years ago, the Johnson Family Foundation gave me a grant to bring together a number of folks, including at least one person sitting in this audience. And we looked at hundreds and hundreds of papers with the question basically being, could we actually start to get any sense about whether a wide number of diseases that actually we see in women presenting to a clinic, whether any of those are actually associated with adult exposure, let's say, versus this concept that we're now talking about of embryonic or fetal origins of adult disease. I think that thing that surprised all of us there, we all had a sense that some things probably had a basis, but if you look at many of these diseases that I've listed here, things like endometriosis and uterine fibroids, pretty universally, it has been accepted and still is accepted by some medical professionals that this is largely a disease of adult women having to do with a genetic defect or something going on in adulthood, instead of thinking about the fact that, as we were able to show from the literature, that many of these seem to be being set up during embryonic or organogenesis. So that sets the context tonight of what I'm talking about. And I think the interesting part is that there's also a theoretical construct that's being developed, which dramatically changes our concept of health and disease. Largely, if you think about health and disease, and I give public talks, I have this little fancy thing where I say, okay, what causes disease? If you go to the local grocery store and ask every lady coming out or every guy coming out what causes disease, they tell you germs and genes, right? I mean, that's the premise. Pathogenic organisms and some kind of genetic mutation. But most of us realize that there's probably not going to be a gene for Alzheimer's, that in fact there's only 28,000 genes or so in a human, which means that more we have to start thinking about genes in the context of language, in which genes put in different order, heterochrony, genes turned on and shut off at different levels, may in fact lead to predispositions in disease, which then later, depending upon lifestyle exposures and Lord knows what else, basically leads to some kind of disease or health. And so today our concept of health and disease is not just what you inherited from mom or dad. You can't just blame them, okay? You can, but it doesn't do any good. The fact is that the environment that your mom and dad, quite honestly, saw in their germ cells, coupled with what you saw during development, coupled to what you're even seeing now, actually is part of this whole concept now of health and disease. And that's very, very different than the genetic determinism that we've been thinking about for years of specific mutations or even groups of mutations leading to disease or pathogenic organisms. So when we actually think about health now, and many of these things are obvious, we actually not only think about health of a developing child as being your parental genetics, or even, let's say, epigenetics now, but we have to think about all these other variables. We've always known that diet and nutrients were important. We've actually known that things like stress and various body burdens of contaminants, exposure to radiation, et cetera, would actually lead to some alteration of this baby. And of course, we have enough crazy, and in fact, crazy pharmaceutical studies, whether it be the DES daughter or thalidomide babies, et cetera, that we actually know that drugs can actually influence developing offspring as well. What hasn't been appreciated, and I think is still being appreciated, and I put this as a placeholder, and calling them personal care products, is that there's this wide array of chemicals that are in the environment that we are exposed to every day that are not designed to actually influence us, but they do anyways. And so that's part of the story tonight. Now, this story isn't just one that you find in the medical literature. It's at the front page of places like Time and all the rest. And in fact, they've even started to explain the concept of epigenetics to folks. And I think this is critically important. The take-home message of this story really is that if you start off bad, then it probably lasts a lifetime. And the story that's actually growing is that every single exposure is a 3-generation exposure. In other words, not only are you exposing yourself, and not only are you exposing the pregnant child, but you're exposing that child's germ cells. So in order to actually do truly multi-generational studies, you have to get beyond the F2, you have to be at F3 or F4. And so that's a concept which also is changing what we're thinking about multi-generational and transgenerational effects. So we all know this. We know that kids are different. I mean, I call them little adults. Unfortunately, for a very long time, we've kind of treated kids as little adults in medicine. But we don't do experiments on pregnant moms, or at least we hope we don't. And we don't do experiments on kids. So we have in the medical community, or even in the biology community, for a long time, developed the concept of a model species. And model species allow us to do things which, quite honestly, we can't do with things like humans. So there's a reason why we have a sophisticated toolbox for fruit flies and mice and worms and various other things. And what I'm going to argue tonight is that there's another kind of model system we should be thinking about, which we use all the time, is something called the sentinel species. And what's interesting is that NIH is starting to bring this into the fore. Things like NSF and NOAA and NIST and a number of other major governmental agencies already do this. But the idea is actually to take species which are actually living in the real world around us, and this doesn't just include wildlife, this also includes things like pets, so dogs and cats living in the home with the same people. So you can actually start to get some idea of what happens in real populations with real genetics that are actually having real world exposures, which are chronic and variable. Now, I put this slide up because actually today, I hope, I wasn't in my lab, but they were supposed to. We have projects going on in every one of these species that you see up here. So we work on a wide array of species with colleagues all around the world, and today, primarily tonight, I'm going to talk a little bit about working on American alligators. Now, the important part of working with a sentinel species is the same concept that working with a model species, and that is you have to be able to interpret the data. And so these two guys, we all know the story of the canary in the coal mine, took it down to test for methane, and these guys are saying there's, of course, something wrong here and I don't know what. That's the same with what we have to do. So if we're working with unique species, we have to understand normal. Even if we're working with model species, we have to ask which part of what we're learning is transferable and which part of what we're doing is not transferable. So that's critical. And any species, you saw the cartoon of the woman with all the different variables. Any wild species also has its own developmental matrix. And I think the important part is that we spend an awful lot of time thinking that we know all of the critical components of an experiment. We think we can actually just control photoperiod or whatever. But the fact is the animals are actually smarter than most of us most of the time. And so these variables do play a critical role in embryonic development. And these are things that you have to take into consideration. Now in my lab, because I work on wildlife, things like climate change and stress and diet and contaminant exposure all play critical roles in basically embryonic development. So tonight I'm going to talk a little bit about endocrine disruption. This was actually a concept that was brought forward in the early 1990s. It was thrown out as a hypothesis. A number of us got together. It was actually a really interesting and weird mix because there were physicians as well as basic scientists, evolutionary biologists, ecologists, et cetera. But what we started to realize is that all of us were working on basically the same problem. We were actually looking at species, including humans, exposed to an array of contaminants, and we weren't interested in the toxic or lethal aspects of these chemicals. We were interested in what happens when you're exposed to a weird mixture of chemicals at an environmentally relevant concentration. So not high enough to necessarily kill you. Does that actually matter? And the fact was, as we started to realize it did matter, and over the intervening, about 18 years now, it's pretty well documented that you can disrupt the endocrine system at every one of these levels, and this is of course a very little simple cartoon of the endocrine system, just a higher level of organization versus the endocrine organs versus the target tissues, and then of course clearance and storage in the liver. But we've now documented that there are chemicals that we are exposed to every day that actually have an influence at every one of those levels. And I might add that 2 weeks ago, the World Health Organization just put out about a 400-page report examining hundreds and hundreds of papers in human health as well as wildlife. And they, in fact, concur that not only are much of these things taking place in wildlife, but they're also taking place in humans. And the data is now starting to become in place for that. And that document is actually online and easy to get. And if somebody's interested, I can pass that website along to the center so that that's available. So the take home is that endocrine disruption is real. That's fine. The question always becomes, but at what level? What exposure? There's all these kinds of questions. I put up this slide only because it's an official slide. I always like to point out that this actually is an NIH slide, and they're almost as conservative as the CDC. So if this thing comes up, they probably have reasonable data. This comes from the National Toxicology Program, but these are the general classes, and quite honestly, to date, they've actually identified over 1,000 different chemicals that actually have endocrine disruptive potential. So this is no longer a hypothesis. There are still hypotheses being generated, but it is a reality that there are chemicals we're exposed to that influence the endocrine signaling system. I should add that this is broader than that. It's now been shown that there's also chemicals that modify cytokine signaling, so the immune system. There's chemicals that modify neuroendocrine signaling and neurological signaling. So we're starting to say that this is kind of like signal disruption versus saying that it's just within the realm of the endocrine system. So how did a Colorado mountain boy end up playing with alligators? Well, in 1985, I got hired at the University of Florida, and I was actually working on high-altitude pregnancy and the evolution of the placenta. And I think I actually even gave that talk here at one time a number of years ago. But, and we still play with where does the placenta come from and how does it evolve its endocrine action. But I got a knock on the door about 6 months in. There was a guy sitting there in a uniform, and he said, "We want you to come and work out the reproductive biology of the American alligator." And I started laughing, and he wasn't sure what I was laughing about, but I said, "Do you realize there's like 100,000 people working on human reproduction today, and they still haven't figured it out? What do you honestly think we're gonna do?" Me, okay, one person doing the American alligator. He said, "Well, we can do a lot of stuff." And we could, because at that point, this animal was still an endangered species. It still is an endangered species, but it's Schedule 2, not Schedule 1, which means the population's come back somewhat. And we have learned a lot, and hopefully I'll show you a little bit about that today. But one of the first things we started doing was go out on warm summer nights, and you'd count alligator eyes. So all these dots are alligator eyes. And what was really nice about this is that you could actually determine the population structure. So how many hatchlings did you have versus how many juveniles or teenagers in the population? How many adults? And you could start to put that population together. And one of the things we started to realize was that recruitment in most of the lakes we were working on was quite poor, because we could actually fly and survey nests, we could survey adults. We started to realize that reproduction was not very good. And so we set up two teams, the innies and the outies. The outies were the wildlife biologists, and they did the skin out. And then my team was the innies. We did the skin in. So my graduate students were required to hang off the front of the airboat. And as we ran up on the alligators, they had to make a snap decision. If the animal's 5 feet or smaller, you grab it with your hands. If it's over 5 feet, you noose it. And the joke in the lab, make a mistake, your dissertation takes twice as long to type, okay? So I actually honestly have to say I can't say we've never had alligator bites. I can say that no one is ever missing fingers, so they're still doing a good job. But the take home is that we started to realize that we really did have poor recruitment. And so if you have poor recruitment, you go to the unit of recruitment. The unit of recruitment is, this is an alligator nest, about 6 feet. I figure you have to have a little bit of biology at this talk, not just molecular biology. Because global warming, I know it doesn't feel like it today, but alligators could be here in the next few years, okay? So you need to know this. So here's an alligator nest, about 6 feet in diameter, about 3 feet high. There's a guard pool around it. This is actually the trail. This is where you send your students in. Remember, students are a renewable resource at a university, right? So this is especially undergrads. Once they're graduate students, they're too well trained, so you don't want to lose them. But the take home is, we come in here, mama actually does defend the nest, so in many cases we can catch mama at the nest, get blood sample, urine sample, tissue biopsy. Same thing happens when you go to the doctor. We do the same thing with these guys, so we had to develop all these techniques over the last few years. We collect the eggs, and we bring the eggs back into the lab. Mama will lay about 35 to 50 eggs. They're about twice the size of a chicken egg. They're hard like chicken eggs. You clink them together, they sound like fine porcelain cups, okay? So you bring them in the lab. You incubate them under controlled conditions, removed from predation and flooding and all these other kinds of things. You say, how good are these eggs? And what we actually started to realize was that even on our best lakes, we were getting 50% hatch rates and 50% mortality. Now, I knew that that wasn't what it was supposed to be, because a few years before this, I actually started working on the Nile crocodile in the Okavango Delta in Botswana, in southern Africa. And there the hatch rates were 85 to 90%. And 85 to 90% hatch rates are probably fairly universal for most wildlife, except for things like fish and all, which broadcast thousands, in some cases millions of eggs. So we knew that there was something going on, and eventually we found a lake, Lake Woodruff. Lake Woodruff is the best, as we all know, right? So Lake Woodruff, in which we had about 90% hatch rates. And so that has become our reference site. And it's important for all of you to recognize this as I start flying through the data here in a little while, is that we define this lake as normal. We have no idea whether this lake is actually like it was in pre-Columbian times, okay? But we have healthy population, they look robust, they lay great eggs, they look healthy. So we call that our reference lake, and everybody else is related back to it as the reference population. So here are these populations, just to give you an idea, and I can't see the map very well, but basically these are in central Florida. So we actually have Lake Woodruff we've talked about. We have another lake called Lake Apopka, which has become famous for its contaminants, and I'll show you some of that in a minute. But it's all relative. I think it's important to recognize, I gave a similar talk to this about 10 or 12 years ago at the Karolinska Institute in Stockholm, and at the halfway through the talk, the guys in the back were talking, and I finally said, okay, what did I say? Who did I insult? And they said, no, we're kind of freaking out because your Lake Apopka is what we're hoping to restore the Baltic to. And so it is relative. So there is contamination, but it's relative, okay? We also have a major study going on at Kennedy Space Center, and I'm not going to talk anything about that today. I should, but I'm not. And then we actually have a whole series of studies now going in South Carolina where I'm based. So here's the take home. You can go to sleep or leave right after this. This is all the data. It's really sad when 10 years of data can be put on one slide. But what we've actually found is a whole series of things, like I've already talked, on depressed hatch rates. I'm going to show you some altered gonadal anatomy that's consistent. Consistent with that is altered steroidogenesis and altered gene expression. The important point is when we raise these animals up in captivity for up to 3 years, or we go out and study the teenagers in these lakes, which are actually 5 to 10 years old, and now we're doing adults, what we find is that the problems that I've described or will describe are persistent. So this isn't baby fat where these guys kind of grow out of it after a year. These are persistent problems. And we've now been, in fact, documenting them, especially on Lake Apopka, which is the main lake I'll talk about that's contaminated. We've now been doing this for almost 20 years. So yes, 20 years sounds like a long time, but for an alligator, let me put something in perspective for you. We marked animals almost 32 years ago as a start. We are now recapturing those same females laying eggs now 33 years later. So last year we caught 2 females, 8.5 feet. They were 8.5 feet long. 32 years ago, we caught them last year. They're 8.5 feet long. They're still laying eggs. So we have a reproductive lifetime that probably is at least 35 to 50 years because it takes 12 to 15 years for those animals to reach sexual maturity, which is 6.5 feet, and then they continue to grow for another 10 or 12 years. So we're talking about very long-lived species. We're talking about species that lay 35 to 50 eggs. So that gives you a little bit of context to try and understand what's going on here. So 20 years isn't even the reproductive lifetime of one female. I know granting agencies believe that we should figure it all out in 3 years, but for some species, that's not the way it works. So when we found these problems, we started looking at all kinds of things. We did fairly sophisticated population studies, population genetic studies, no differences. We actually looked at diet, no differences. We looked at microenvironment, gas environment of nests, no differences. We looked at moisture differences, no differences. We did primitive temperature. We're now doing sophisticated temperature analyses of these nests, no differences to date. What we did start to find differences is not the kind of fish they ate, but what was in the fish they ate. And so when we started to look at what was present, what we started to find were a series of heavy metals and a series of contaminants. Now, all this is just a stacked bar graph. This is this is actually part per billion. And part per billion, quite honestly, up until about 10 years ago, was almost non-detectable. Now we can detect it. And if it couldn't detect it, therefore it didn't have any biological action. And of course, we couldn't detect hormones either for a long time, and we know they have a few actions. So let me just give you a scenario here real quick. We've all heard about DDT. In fact, there's still a raging debate going on about once a year, there's an editorial that goes to the New York Times saying we should bring DDT back, okay? 1952, DDT was shown to be estrogenic. If you overectomized a hen or you overectomized a rodent and you injected DDT, the uterus proliferated. It's a gold standard for most estrogens. And in fact, it's been documented over and over again that it's perfectly estrogenic. So orthoperic DDT has been known to be estrogenic for more than 50 years. We also know it's associated with premature cessation of lactation in women who are exposed to it. We know it's also associated with an increased level of miscarriage and premature labor. So there's a major argument going on now in the World Health Organization, in which I'm part of, in which we're saying if you're a tropical disease doc and you're interested in malaria, DDT is a magic bullet still. But if you're a basically reproductive or OBGYN and you're worried about babies surviving in the third world, premature cessation of lactation or premature birth is probably a death sentence. So there's a real concern of why are we still trying to use a 50 or 60-year-old technology, that is DDT, when there's other solutions that may in fact be better. That's my end of my political statement today. So that's a different story. But here's two quick things. DDD is one of the major metabolites of DDT. It's sold in this country under a trade name called mitotane. It's actually used to treat adrenal cancer. It blocks adrenal steroidogenesis. It's still used in the vet world for the same thing. So here is a metabolite that's actually detected in most people that actually have been exposed, which means most of us, because even though DDT is no longer used in this country, it's still extensively used in the tropics. It still comes in foodstuffs, et cetera. And DDE is the metabolite of DDT that's been detected in every single human sample that's gone through the Centers for Disease Control in the last 25, 30 years. It's an anti-androgen. It's about as potent as hydroxyflutamide, which we actually use or used to use for prostate cancer, but now it's used as an experimental anti-androgen. So this is just to give you an example of a couple of compounds that are metabolites of DDT. So we're shocked that there are endocrine active compounds in the environment, but the point is a number of these compounds we've known for 40 or 50 years. It was considered to be a dosing issue. If you're worried about toxicity in this classic sense, lethality, then few of us are exposed to toxic concentrations. But if you're interested in biological response, that's a different story. Now the take home is just because you have contaminants and just because you have problems, that doesn't mean that one causes the other. All you have is an association. And so the question then becomes, can we start to show that some of the problems that we're having in these populations are more than just an association? Can we start to get to causation? And what I'm going to do tonight is focus on the embryo and the developing egg, and we're going to focus on the ovary to start. And some of this is not new to some of you, certainly not to at least one person in this audience, because she actually helped collaborate on some of this work, and that's Teresa Woodruff. And so if you actually look at the developing ovary of an alligator and you actually take a biopsy and you slice it through, this is what you find. Nice ovarian follicle, single nucleus, Balbiani body, and basically granulosa around it. And if I take blow-ups of this and I slap a mammalian ovary up there next to it, it's not a Graafian follicle, but an early primary follicle, you basically can't see the difference, okay? In contrast, if we actually look at our animals from this Lake Apopka, where there are lots of contaminants, poor recruitment, et cetera, I asked a simple question, and the question was the following. If a female lays 35 to 50 eggs every other year for 50 years, and all she has to do is produce 2 offspring to reach sexual maturity, and the population's stable, right? It's 0 population growth. Then who cares if 50% of the eggs die? I mean, of course I care, but I mean, who cares from an evolutionary sense if those die? So the question then becomes, are the little guys that hatch out, are they healthy? And that's why we started to look at biological responses, because the fact is if the female that survives, or the male that survives, is for some reason reproductively compromised, then they're filling a niche, they're filling a hole, but they can't produce the next generation. So you in fact have a problem with recruitment. So we started to look at these eggs, and in this case, when we looked at Lake Apopka, what we actually found was a condition called the multiocytic or polyovular follicle. And of course, this didn't get its name because Lou Guillette found it in an alligator. In fact, the original description of some of this was actually in mice and in human females that had been exposed to DES. This is part of the DES story or the DES daughter's legacy. For those of you who are young in here and don't know this story, late 40s to the early 70s. It's a guess, we think about 7 million. Some people say only 3, but some people say 12. And so we average it and say, well, about 7 million pregnancies were treated in this country with this synthetic estrogen called DES. It was given for premature labor. It was given with incredibly crappy data support. If you are interested in the story of some of this kinds of things, there's actually a pretty interesting book called Toxic Bodies that was published last year, which goes through basically the whole history of this from both the political end of it, and it's quite an easy read, but all I can tell you is it just made me madder and madder, because when I actually testified in Congress in the 90s, I thought I was presenting new stuff. And it ended up that almost the same stuff, different words, different data, was presented in, the 1940s by scientists who were my, if you will, ancestors as reproductive biologists. And it's been done every decade. Seems like the same things are said. Neither here nor there. The take-home is that this multiocytic follicle has largely been associated with an increase in infertility or subfertility. And some of the work that's done suggests that these eggs can be fertilized and can be taken to viable offspring. But in some cases, it's associated with early miscarriage or loss of young. So this is actually an older slide. I don't have all of Kelly's new stuff on here and others, but the context is the following, is that if you, in fact, disrupt estrogenic signaling, and then all of the things that are associated with it, you lead to multiocytic follicles in an array of species. So not just alligators and caiman, but mice. And you can even generate this by giving soy compound like genistein that was actually done at NIEHS. So our hypothesis was that the mortality we were seeing and all the problems that we were seeing were not due to the toxic effects of these contaminants on the developing embryo. It had to do with abnormal ovary development. So let me just run through a couple of questions and try and characterize for you. When you work on a species that's a non-traditional species, you basically start at the beginning. And you can't assume that just because it was found in a mouse, you've actually going to find the same thing here. So you actually have to go and do the work, which in some ways is fun. In some ways it's a pain, because you actually have to start and build all the tools from the beginning. So one of the first questions we had to ask is, how do you get an ovary? I mean, how do you get a testis? What about what about sex determination? Well, alligators actually are wonderful from one sense, in that depending on what temperature you incubate them on, you get either females or males. So I can take all 35 or 50 eggs and make them all sisters by incubating them at 33 degrees. And all I need to do is do that for about 72 hours out of the whole 2.5 month period. So it's a tiny little window, sensitive period. It's thermal sensitive. 30 degrees, 100% females. 33 degrees, 100% males. So I can actually design experiments where every well of the experiment is full of sisters, so which makes it fairly nice, and actually NIH has agreed and funded this every now and then. But the take home is that you can also do an interesting thing in that you can incubate at a male temperature, add an estrogen, and only estrogens, and you can redirect the sex. So you can actually turn it into a female, okay? So kind of a cool system. And that's why we have different kinds of models, because each one has a strength and they have weaknesses. So what do we know? We know, for example, in that developing ovary that things like anti-Mullerian hormone, DMRT1, SOX9, aromatase are all expressed just like you see in any other gonad. And if you're an ovary, you got boatloads of aromatase, and if you're a testis, you have boatloads of SOX9, okay, and anti-Mullerian hormone. So the system seems to be, even though it's actually temperature-induced, the genes involved are very similar, if not identical, to the same ones we see in mammals. And of course, aromatase is this enzyme that converts testosterone to estradiol. And in fact, if you give increasing doses of estradiol, you increase the percentage of sex. So I told you sex reversal. So it works. In fact, it works down to 100 parts per trillion, which is picomolar concentrations. Okay? So we're talking about exquisitely sensitive system. Let me explain this experiment to you really quick. You basically take an egg and you put a drop of alcohol with whatever you want to deliver on the outside of the eggshell, and it goes through the eggshell and is picked up by the chorioallantoic membrane and delivered to the embryo. If you crack the egg or open it up, then fungal infections and all that things take place, so you basically kill the embryo. But if you put it on the egg, it's nominal dosing. We know that probably, depending upon the polarity of the compound, 50 to 60% gets in. So these are actually nominal doses is what we put on the eggshell. It gets in, delivered to the embryo, and you get perfect good sex reversal. The other interesting part is that we stage these at stage 19, which is actually a stage 2 before sex determination. So it's about 3 days before actual sex determination takes. We put it on, and we only do it one time, and then it'll get sex reversal, okay? So this is incredibly, exquisitely sensitive. And what we know now is that one of the things that's going on to actually make an ovary, and I just throw this up as one piece of a huge data set that we've actually gotten, is that if you actually look at the aromatase gene and you look at the promoter, there are in fact 6 CpG islands, and if you're in fact a female, you have hypomethylation. If you're a male, you're hypermethylated. So in fact, 5 of these sites out of the 6, you have a significant difference. So again, this is actually the first demonstration that methylation works in a reptile. Why wouldn't it? We've actually found it in fish and we found it in mammals, so it's be really shocking if it didn't work in a reptile. But the fact is that aromatase is being shut down by methylation in males. So we're starting to understand what's going on there. We have a whole bunch of similar data for SOX9 and AMH regulation. So we have a system that looks somewhat similar. The question is do they have estrogen receptors? The answer is, well, we have to go in and actually ask. And sure enough, there's 2 estrogen receptors in alligators. They're basically found in chromosomes 1, just about like in every other species been looked at. And if you look at the structure, they're very similar to every other steroid receptor. But the 2 interesting things, if you look at the DNA binding and you look at the ligand binding sequence similarity, they're 100% similar between alligator, mouse, and human. What that tells me, and I think this is an interesting evolutionary story, is that if you have any mutation in your DNA binding region of the estrogen receptor, you are not represented in the next generation, okay? In other words, this is lethal as far as reproduction is concerned. You're just no variation. The variations exist, for example, in other regions. Even the ligand binding region is highly conserved. So the sequence is about 95%. We've now sequenced Tai Eguchi's lab in Japan and my lab. We've now done, I think, over 50 species of estrogen receptors. We've done 20-some fish. We've done 6 or 7 sharks now, et cetera. Interesting thing, sharks only have ER beta, which is kind of cool. They don't have an ER alpha. But the fact is that this kind of similarity, or in fact, strong conservatism exists throughout, especially for the DNA binding region. So we do have an estrogen receptor. Sequence-wise, it looks like an estrogen receptor. You can transfect that via plasmid. We have a couple of different systems we work with in the lab, and I'm not going to belabor this. You add the hormone, you add the plasmid, it's a reporter. If in fact it's an estrogen or an androgen or whatever receptor you put in, you basically can measure, in fact, gene expression as luciferase by transactivation. And sure enough, all of the estrogens, estrone, estradiol, and estriol, all work really well, and they all work really sensitive, including the interesting part is that snakes, I think, have the most sensitive estrogen receptor we've seen so far. They literally work at 10 to the minus 13 in this system, so exquisitely sensitive using natural hormone. And you can also do contaminants. And so, for example, you can take things, for example, like transnonachlor, which I haven't talked about, but is actually a very common estrogen, part of chloridane. If you live in the South, almost everybody has chloridane in them and transnonachlor because it was put in the foundations of almost every house that was built in the 60s and 70s for termite control. So it's a very persistent organochlorine. And transnonachlor is a reasonably good estrogen. Interestingly enough, it's called a weak estrogen because it's about an order of magnitude or 2 less, has a lower affinity than estradiol 17 beta. But if you get the right dose there, you get 100% gene expression. And sure enough, we've shown that. So there's this problem with calling a compound weak just because its affinity for the receptor is weaker, I should say. It still gives you the same activation. So you can put different compounds and mix them together, and you can do sex reversal. So all of this is just taking alligator egg, putting the compounds on, single dose, and sure enough, you get sex reversal. And so these are real estrogens because only estrogens do that. They're weird estrogens. They don't give us the normal dose response that you see with estradiol 17 beta. So something else is going on molecularly. I mean, it could be, in fact, the cofactors, and we're spending a little bit of time trying to figure that out. But the take-home is that, I mean, they are exquisitely sensitive, 100 parts per trillion, and we actually get sex reversal. So what we did was we went ahead and took eggs from the contaminated environment. We incubate them at a temperature. I didn't say this, but if you do an intermediate temperature, 31.5, you get half males and half females. Not hermaphrodites. They're half males and half females. So there's a genetic basis for sex. It's just overridden by temperature. Okay? So the interesting part is, sure enough, if you take the animals from the contaminated environment, you get 76% females. So the assumption, of course, instantly, at least my students' assumption instantly, was that we were sex reversing animals because of contaminants. There's a problem. The problem is that in the yolk are also natural steroids. And so if you actually think about follicular fluid, which is usually not very well characterized, it ends up when that ovary is making all that stuff that it's dumping into the bloodstream, it's also dumping it into the follicular fluid. In this case, it's dumping it into the yolk of the alligator or the bird or whatever. And you can measure all kinds of hormones present. And in fact, those hormones are mobilized by the embryo and they move. This is actually just looking at early and late is just before and just after sex determination. So you can actually see that hormones like E2, progesterone, and testosterone all are reduced in the yolk during that process of sex determination. So what's the influence of native hormones? We're still trying to figure that one out. It gets even more complicated. And this is actually, I put this in for anybody here who's interested in evolution. So, placenta, right? Endocrine placenta, uniquely mammalian? Nope. Reptiles actually probably had it long before mammals did. In fact, the first mammals were egg-laying, and they were probably live-bearing reptiles before there were ever live-bearing mammals. But the take-home is, we asked a simple question a couple of years ago, one of my students and I, and that is why can we assume that the chorioallantoic membrane that gives rise to the mammalian placenta in a non-mammal, how can we assume that it's not endocrine? And so we actually went in and looked at a lot of the enzymes. I just put up two, a lot of the enzymes associated with making steroids, and sure enough, they increase. In this case, this is a chick. So this is something that's been studied for God knows how many years. It ends up the chorioallantoic membrane makes steroids. Specifically, it can make progesterone and has progesterone receptor to receive that signal. So the take-home is that we also have, as well as yolk steroids, we actually have a embryonic membrane, the chorioallantoic membrane, that literally can convert steroids and synthesize steroids de novo. So when we actually think about this developing embryo, we like to think that the reptile or something like that's simple because we don't have viviparity and we don't have a placenta. We have to think again. So this goes back to this idea of complexity. It's not as simple as what we think. So last part of the story, why study baby alligators? And this is one of the reasons. Of course, mice also are an advantage. In the human female, when that baby girl is born, every ovarian follicle, this is the dogma. There's a little bit of argument out there right now, but the dogma is that every ovarian follicle she's ever going to have is in that ovary when she's born. In fact, she's already been going downhill since about month 6, right? Now, she still has millions, and not many women I know are interested in that kind of fecundity, so that's actually okay. But it's difficult to try and study the human ovary when all of these processes are taking place inside of mom, even if we wanted to study that. So in fact, folks here have used a mouse model because a lot of that takes place in the first few days. And the cool part about the alligator is that when it is born and it hatches out, it is still synthesizing oogonia. It still has oogonial nests. There's still massive mitosis going on of the oogonia. This is actually a 3-month process to go from here till we get the first follicles. And so we've been able to study that process also a little bit longer than it takes in a mouse, but we actually start a little bit earlier and we end up about the same. And in collaboration with Teresa Woodruff, we started looking at inhibins and activins because work that she and Kelly had done here clearly demonstrated that it played an important role in follicular genesis, but the comparative part was still poorly understood. And in fact, a little side story I told the folks at lunch. So there's a class that's taught at Woods Hole every summer, Frontiers in Reproduction, and Teresa invited me up to give a seminar, and so we went up there, and then of course you have to go afterwards to the Captain Kidd, and it's loud enough that you have to scream at one another to try and talk, but we started talking about inhibin, and she said, you know, we just cloned out, I think, chicken inhibin, and it's weird, it's unique. And I said, why do you think it's unique? Why maybe mammals are unique? And so we hatched a program that night, And I think in the process, we cloned out over the next year or two, I think about 12 or 14 or 15 different species. And sure enough, it was the mammal that was unique. Most of the other vertebrates actually had the piece that was missing that the chicken had. But one of the things we started to ask was, could we actually show that these were universal growth factors in reproduction, again, across vertebrates? And so the take home is, do these factors actually play a role in ovarian development in alligators? And I just put this up to represent a whole dissertation that was done by one of my students. But the take home is yeah, all the same factors, the inhibins, activins, follistatin, everything that you actually see in the mammalian ovary is also present in the alligator ovary. And it shows the pattern, which is actually pretty similar. But the question we wanted to get at was, were these gonads functionally different? So the downside of an alligator, as I just told you, is it takes 12 years to reach sexual maturity and they reproduce for 30 years. So you're not going to do multi-gen studies on an alligator. So one of the questions we wanted to ask is, could we, however, challenge these ovaries and determine whether, in fact, the problems that we would perceive later in life are already present? Could we already identify them literally at the beginning of life? And so we actually did FSH challenges for animals in Apopka and Woodruff. And a couple of really, really intriguing things came out of this work. The first was, remember I told you that it takes 3 months to get ovarian follicle. But if you in fact challenge these animals with FSH, even at 1 month, you actually get estradiol synthesis and you actually get inhibin production. Now these are supposed to be coming from granulosa cells. There are no granulosa cells at this stage. And what's interesting is I presented some of this data a few years ago in New Zealand, and someone ran up afterwards and said, well, we got the same thing in sheep. We're starting to find the same thing in sheep. So I mean, I think that this idea that an ovary doesn't have these signaling molecules until sometime later, I think may in fact be false. And I think there's enough evidence that's starting to suggest that. The other interesting thing is that if we inject Lake Apopka animals, not only do we get an increase in estradiol, but we also get a significant increase in testosterone. So these females, when you're challenged, we're talking about early in life, their ovaries are not only making increases of levels of estrogen, they're making increased levels of testosterone, and that is not normal. The reference population does not show that. The reference population shows you an increase in follistatin and an increase in aromatase, but the contaminated population does not. And if you look at things, for example, like aromatase, SOX9, and AMH, what you in fact find repeatedly is that the contaminated and exposed individual responds differently than the reference population. In some cases, no response. In some cases, an opposite response. So the point here is that these studies are done in the first week of life. There are no ovarian follicles. We're getting responses already from this ovary. We now know where it's taking place. In the hilus or the lacuna region is where, in fact, the synthesis of the hormones is taking place. And so the question then becomes, these are present, these are birth defects present at birth. So let me throw out an idea for you. When a developing embryo starts off, it has an indeterminate gonad. It has a gonad that has the potential to go a couple of different ways. And there's still a little bit of debate about this, but the idea being is that when a germ cell hits the ovary or hits the testis, or if I will say, this primordial gonad, it has the potential to become either oogonia or spermatogonia. Depends upon the local signaling that it gets. The same with things like granulosa and Sertoli cells or thecal and Leydig cells. What happens if during development you don't get sex reversal? That's simple in an alligator. What happens if locally, focal areas of the developing gonad get the wrong signal? What happens if you actually have cells that are present in the ovary that actually think they're Leydig cells? They're not thecal cells. In fish, we actually know that we can do this. We can actually make fish where if we give them low dose exposures to estrogen, we can actually make a fish that actually has perfectly good seminiferous tubules, but it has growing oocytes within the testis. And we've now done the reverse. We have ovaries where we can actually get some of the cords to set up, and you have spermatogonia developing in the gonad. Now, of course, these are fish. Sex determination is a bit different. But that doesn't mean that some of these cells could in fact be reversed. We like to think that everything in the ovary is being affected, but what happens if just focal points are? So the idea is environmental steroids, these environmental contaminants could be altering local signaling, and it's probably much more realistic. We already know that if you expose a developing female to androgens, for example, that's the standard model for PCOS, that you actually get a PCOS-like model, okay? And so the take-home is that although we largely think of PCOS as being genetic, we clearly know that there is a potential for environmental signaling, and in fact, many of the markers we have in our animals are representative of PCOS, or premature ovarian failure. So, this is not unique to alligators. I finish up here by, we have a wide array. In fact, I can give you every vertebrate group, from sharks to polar bears, have been described as having altered steroidogenesis associated with contaminant exposures. These are ecologically relevant exposures. We're not talking about they live downstream from a dioxin factory, okay? So the take-home is that this concept of altered steroidogenesis, altered gonadal development is present throughout vertebrates. Not every species, not every individual of a given species, of course, but clearly these have been described, and it's not just alligators in Lake Apopka, it's not just a few fish in rivers in England. This is far broader than what most people perceive. We actually also wear another hat, and the hat is that we also have a major project that goes on in South Africa. We work on Nile crocodiles. And there's a direct relationship between what we're doing and human health, because where we work, we are in fact are associated with villages where women go down and not only wash the clothes, but this is where they actually take water in buckets and take it home. So the wildlife, the fish work, and the crocodile work we do is a direct exposure correlate. And so there's some funding that we're actually doing with the USAID, but most of this is actually being supported by the South African government, and it's being supported by the University of Pretoria, so we work with them. We've also started a major project on great white sharks, because they're a global species. They're off of South Africa, they swim to Australia, they go down to the pole, South Pole, and then they swim back. They do that all in about 6 or 7 months. Pregnancy is over 2 years. They eat fat from whales. They have high contaminant loads. And what we've actually done is cloned out the receptor, and we're starting to actually use molecular technology to try and determine which contaminants we find in great white sharks may in fact be the ones that contribute the worst to this species. And the fact is that we have many of these sharks that are losing pregnancies that have been documented by the ecologists and the shark biologists that work on them. So this is actually trying to take our modern molecular tools and not only say something about human health, but also say something about the world, about us. So I actually, as you just heard, I actually have a chair in marine genomics, but the medical school in South Carolina, Medical University of South Carolina, is a full partner in a marine lab. Pretty unique. I think Stanford's the only other place that actually has a full marine lab associated with this medical school. And so I'm the director of not only the Center for Marine Genomics, but also the Center for Marine Biomedicine. And this is just a few of the projects that we actually currently have going on just in my lab that we're funded for, everything from oil spills in the Gulf and dolphins to some of the kind of work I've described today, including work with infertility clinics in South Carolina, trying to characterize follicular fluid and granulosa cells for women coming in exposed to various kinds of contaminants in our environment, including discovering new EDCs. So I don't have to make the point here, but if you don't understand normal, you can never understand abnormal. And that's a central issue. It's not just a central issue in being able to interpret data, but we need to understand what's the variance that exists in normal populations. I put this up because it's a great slide. Daisy Bubble and Elrod's just like someone slipping super hormones into their feed. This cartoon was drawn by a guy. How do I know? He has absolutely no idea what the udder is for, okay? So you got a second set of mammary glands. Never took reproductive biology from Kelly or I, ever, okay? The take home is that also you have a chicken with mammary glands. So if you don't understand normal, I can actually give contaminants, for example, to my baby alligators and change sex. You're not going to do that in humans. But the question is could you actually change the fate of cells in the developing gonad? And that's a different kind of question and one that we actually need to address. The other thing is that we all know that the environment plays a critical role in the selection of the genome. That's something we've been teaching for, kind of a century or more. We now know that we also have an epigenome. And I think the important part that we have to basically really take into consideration when we think about health and disease is that we actually are modifying the genome by the environment. And so this environment no longer is this little tiny thing that, we just kind of worry about putting on sunscreen and not smoking. The fact is the environment is playing a much bigger role in our health than we actually, I think, ever dreamed. So the final take home is that I've shown you, or at least I hope I've shown you today, stuff that goes literally from population and organisms to molecular. I actually have a team, and I'll show you the picture here at the end here. But I think that this call for multidisciplinary, cross-disciplinary research is not just kind of make-believe. And this is now preaching to the students. Modern science and future science, if we are going to understand environmental health and disease, this is not going to be done at just the molecular level. And it's also not just going to be done at the epidemiological level. We're actually going to have to work together in teams. And I'm lucky in that I have world-class chemists and world-class statisticians and physicians that are all work together with me to try and, in fact, address these questions, because there's no way in the world I can do this by myself. So I basically stand up representing a whole bunch of people. And it always takes money, and it always takes time, and it takes talent. There's a number of artists. Hopefully you've enjoyed the slides. But what we do in my lab is to try and understand how she generates one of these guys. I think if the environment is not healthy for her offspring, it's not healthy for ours. And our goal is not to be anti-anything. Our goal is to be pro-health. And so we want healthy babies, we want healthy grandkids, whether they're our grandkids or whether they're, in fact, her grandkids. So I thank you again for inviting me. I'd be glad to answer any questions.
Dr. Guillette will take questions, but before we do that, I have just a couple of announcements. First, we'll take a number of questions. The lecture's a few minutes late, but that's great. We enjoyed it. We won't have a long question period, but you'll be able to talk individually and in groups with Dr. Guillette outside. We have various refreshments and so forth. The second thing before we take questions, I have to thank a few people who were involved in this. So first, I'd like to thank the Center for Genetic Medicine staff, particularly Michelle Moni, who put this largely together, and the rest of the CGM staff, and Dr. Peter Kopp, who's the director of the Center for Genetic Medicine, and again, the Herman and Bea Silverstein Foundation for making this possible, and finally, Dr. Guillette again for his terrific seminar. And I will ask that people ask questions on mic. Using the microphone. This seminar is being taped, and if the questions aren't spoken into the microphone, then they won't be on the tape, and we won't know what's happening. So please use the microphone. Michelle will be wandering around offering the mic to people with questions.
Any questions? How to catch an alligator?
It was a fantastic talk. Really enjoyed it. Are you seeing any other effects on wildlife, like obesity? Are you seeing any body weight changes of any sort that there's either causality or association?
No, that's a great question, and it's a great question for a couple of reasons. And this goes back to this what's normal. So we are not seeing obesity in the alligators. In part, they don't have subcutaneous fat like mammals do. And so obesity in that sense probably doesn't work, but what we're seeing is changes in body proportions. So we're getting differential growth. In contrast, we're doing some stuff on dolphins, and there we are seeing differences, and we're seeing actually a dramatic increase in obesity. And this is crazy. This is a wild animal that basically is dependent upon swimming every day to catch its food. So it's not sitting in front of the TV, eating potato chips. And so seeing any kind of change in metabolism, seeing any kind of change in body composition in a wild animal is, quite honestly, it was, I made the prediction that absolutely we'd never see this because of what I just said. The fact that we're starting to see it actually is kind of surprising. We've also seen, and I didn't talk about it tonight, one of my PhD students, Ashley Boggs, did a beautiful dissertation. We're showing a number of effects on the thyroid. And so we're actually finding that a number of these guys are coming out hyperthyroid. They have goiters already at birth. And that's actually associated with some of the stuff we see where there's heavy metals. And we are doing a fair amount of stuff on obesity as well. So liver effects, we've also seen liver effects. There's an enzyme called CYP3A, which is actually essential for steroid metabolism as well as pharmaceutic metabolism. It's sexually dimorphic. Most people don't know that, but males and females are supposed to be expressing this differently. If you're exposed to contaminants, especially contaminants like estrogenics or anti-androgenics during development, you lose that sexual dimorphism, and it's usually the female that actually loses. So they become more male-like. This is important because this is the enzyme that drug companies design drugs to be metabolized by CYP3A. So an alteration in its activity potentially has effects on even how we metabolize pharmaceutics. That's a good question.
Your talk focused on ovarian function. What about testicular function?
Yeah, I picked on the girls tonight, and I apologize. I wasn't introduced tonight as the alligator penis guy, but I have been in various places. We have demonstrated that there are effects on the testis. We're actually in the middle of doing a fairly large study of adult males now, and I will not go into detail about how we collect semen samples. But we have in fact shown that males on the contaminated lakes actually have small phalli. It's present at birth, it persists, and so we've actually, I could have given a whole nother part of this lecture on hypospadias. We also spend time working on hypospadias. Hypospadias is a very common abnormality of penile development. Instead of the urethra coming out of the tip of the phallus, it comes out somewhere along the underlying shaft. We have both a baby boy study underway at the medical school looking at this, as well as doing wildlife studies. And so we have documented abnormalities in genital development, and we've documented abnormalities in testicular development, but I didn't focus on that tonight. They only let me talk about that every now and then because now I'm a professor of OBGYN, so.
I realize this isn't your area, but do you look at all at differences in behavior between sexes?
That's a great question, and it's not my area. And one of the things I have is I have a graduate student almost every year apply to me wanting to do behavior, and I say, behavior is so complex I can't even figure out what I do most of the time. So we actually did do behavior, not on alligators. We've also worked on fish in these lakes, especially mosquito fish. They're small viviparous fish. They have multiple generations and they have very distinct behaviors. And we've actually been able to show that we have altered behavior, feminization of males and masculinization of females in mosquito fish from these lakes. So that's a good question. We've never done gators, in part just because they'd be incredibly difficult to try and study. They take large areas and they don't really behave normally in an aquarium in the lab. You'd actually have to try and do this in the wild, and that would be really difficult. But it's a great question.
Do you have a hypothesis about what's going on at Lake Apopka that you're seeing these effects?
Yeah, I'm sorry. You know what? I just realized when you asked that question that I forgot a whole little piece I should have told you. So Lake Apopka is the 5th largest lake in the state of Florida. Right after World War II, they took the whole northern part of the lake, which was a marsh, and they diked it off, drained it, took out the stuff, and started muck farming. Muck farming is a traditional southern recipe for, quite honestly, destroying freshwater lakes. What they do is they take the marsh, they actually grow crops in it. Because of the soil types, et cetera, they get a lot of nematodes. So when they harvest the crops, they then take lake water and pump it onto what in fact were the old marshes and flood them to about 12 or 14 inches, leave it there for a couple of weeks, and then they turn around 2 weeks later, take the water and pump it back into the lake. So all of the pesticides and all of the nutrients and everything that they were put on the lake, on the land, then end up in the lake. And they do that over and over and over again, and they've done it forever. So once we started describing the things we were finding in the late 80s and early 90s, by the mid-90s, the state of Florida bought the farms that were on the north end of Lake Apopka, and they turned it over to the Water Management District. And the Water Management District decided that the simplest way to make emergent marsh was just to flood the fields again. So they did, and all kinds of birds came in, and they basically had one of the largest organochlorine die-offs of birds since the 1960s. And thousands of birds died, including a number of endangered species. And so the feds got involved and judges got involved. And they took all that water and pumped it back into the lake, which gave me another 20 or 25 years worth of research. So the take-home is that the lake is largely contaminated from agricultural chemicals, but there are two towns, Winter Garden and Mount Verde, that also use it as a tertiary treatment facility, that is the lake, for their sewage. So we also have sewage going in the lakes. And that's something that I think most people don't recognize is that most small towns in the United States do not have sewage treatment facilities. So they take solids, they basically take the solids out. They have settling ponds, et cetera, but they're not designed to remove all the chemicals that are being dumped in the toilet or washed down the sink or whatever else. So the vast majority of these compounds are not removed, and many of them end up in the waterways. So the answer to pollution, still for most people, is dilution, except we know that doesn't work that way. So I apologize for not making that clear.
So if this problem does perceive in humans, do you think it's possible to fix it using some sort of medication or possibly gene therapy?
You know, that's a good question. And actually, I mean, Teresa Woodruff probably is a better person to answer that than I do, because she's actually spent time really thinking about that. The way I think about it is the fact that if this is not a gene defect in the classic sense, then if we can actually remove a number of these compounds, and again, the way I present this is that this is a predisposition for disease. It's not an absolute. And so if in fact we're aware that these predispositions exist, and we're aware that certain compounds can cause these, then the idea is that through education, we might be in fact able to change some of these things around. Of course, I can't educate my alligators. What I have to do is actually make sure that their exposure is reduced. Now, it's hard for some of the chemicals we're dealing with. I mean, you live right next to one of the Great Lakes. It ends up that they've been monitoring the fish in the Great Lakes for decades. And sure enough, DDT and DDE and all those things have gone down, but the half-life of many of these compounds are 30 to 40 to 50 years. That's the half-life. And if these compounds are working at part per billion concentrations, it's going to take a very, very long time to get rid of all those compounds out of the food chain. And so I'm not trying to be pessimistic. I think there's an optimistic side of this, and that is if it's not classic mutation, there's nothing you can do about that. I mean, there's some suggestion with gene therapy, but I don't think gene therapy is as straightforward as we all thought it was going to be. It's far more complicated because in most cases, these diseases are not based upon single mutations that are easy to rectify. I think that I'm glad to keep answering questions, but I don't know if people want or need to leave, or you want something to eat.
I'm wondering about all the billions of people excreting drugs and metabolites into the water system.
That's actually a great question. And let me tell you a little tiny, really quick story. So they found all these fish in England, below sewage treatment facilities, males that had eggs in their testes. And they tried to figure out what was going on. And they identified a couple of industrial chemicals that are clearly involved. But one of the chemicals they found involved was something called ethinyl estradiol. And what they actually realized is that it's birth control estrogen. So when a woman takes a birth control pill, it goes through her body, of course, and goes through her liver, and it's glucuranated. A sugar is added to it, makes it water soluble. She excretes it, comes to the sewage treatment facility, goes through the bacterial beds, and guess what the bacteria want? They want the sugar. So they cleave off the glucuronide, they take the sugar, and what comes out the other end of the sewage treatment facility is activated ethinyl estradiol. It ends up, it's the same with basically all of our antidepressant drugs, our anti-cholesterol drugs. That's really how we make them water soluble and metabolize them. So they now are measuring, for example, antidepressants and anti-cholesterol drugs in the fish below sewage treatment facilities. So they are there, they're detectable, they're quite low, But the funny part is that you can now go below a sewage treatment facility, and they're doing this in Europe and various places, and you can actually almost exactly recreate the structure of the population based upon the drugs that you're seeing. So, for example, if you see a large amount of ethynyl estradiol, especially a very high percentage of ethynyl estradiol, you know it's a college town. Thank you all. I appreciate it.